So after a tissue-engineered product, we may step back a little bit and come to gene therapies and maybe non-viral vector systems to be used in order to get the transfection made. And for this, we have Dr. Matthias Schumacher from Leon-nanodrugs out of Munich. And thanks, Matthias, for making that here.
Yeah, thanks a lot, Marco, for the nice introduction. And also thanks to Haro for the invitation to present at this fantastic symposium here. My name is Matthias Schumacher. I'm the head of engineering and development at Leon-nanodrugs. And I'm also between you and the lunch break. So I will try to make this a little bit entertaining and hope you can stay with me for the next minutes. Unfortunately, we already had some basic introduction on cell and gene therapy and the concept of mRNA or RNA therapeutics this morning, so I can shorten this a little bit. We see that mRNA or RNA, because there's different kinds of RNA, is a very versatile tool that can be applied in the context of many different potential treatments. Of course, and I cannot go here without the COVID reference, we can think about vaccines for infectious diseases, but we can also think about personalized vaccines for cancer treatment. We heard a lot about cell therapy. There is approaches towards genome editing, therapeutic antibody production, and so on. So a number of different approaches or therapeutic approaches where we can use mRNA as a tool to instruct the body to do certain things. So it's a very versatile tool, and it can be developed rather rapidly if you compare it to conventional technologies. That's plus on the speed and versatility points, but it's also pretty safe. I mean, mRNA as such is a transient messenger in the body. It doesn't integrate in the genome usually, unless we explicitly want to do that in terms of the genome editing. There's different tools, but overall it doesn't come with permanent changes to the patient genome usually. And the cost, if we compare to conventional biologics, it can be also very cost effective. However, in order to have mRNA have any effect on the body, we need to bring it into the cell. We heard about that before. But unfortunately, naked mRNA is not very well taken up by the cells. This has to do with the size and also the charge of mRNA. But also, mRNA is not meant to exist outside the cell usually, and therefore it is digested by enzymatic reactions. It can trigger immune responses and will be cleared by parts of the immune system. And finally, once it has entered the cells, and this is a process that usually goes via endocytosis into the so-called endosome, the escape from these endosomes is usually a process that we also need to influence and to facilitate. And to overcome all of this, we are using nanoencapsulation of mRNA molecules. This allows us to protect it from enzymatic degradation, also from physicochemical damage. It allows us to optimize the uptake into the cell, to tune the escape from the endosome, and also to have it released inside the cell in a pretty controlled manner. And finally, these nanoparticles we build around it can also allow us to target the mRNA, for example, to specific cells. Lots of activities are ongoing in that field as well. So what can such carriers be? There's a number of, I would call it traditional approaches based on viruses, because what a virus does is transporting genetic information into a cell, so you can borrow that system. There's different kinds of viruses, but we can also use non-viral vectors, which in our opinion are a bit easier to work with and also safer. And there is the famous lipid nanoparticles. This is what I want to talk about today, but we can also think of what we call polyplexes or polymeric nanoparticles, different approaches to encapsulate mRNA and bring it into the cell. There are some other approaches. We also heard about that before in context of the Happy Cell Factory, for example, like electroporation. But these are technologies that happen outside the body with these nanoparticles. These we can inject into the body and have the process happen inside. And the traditional lipid nanoparticle usually consists, of course, of the mRNA, the payload, and usually 4 different components, usually an ionizable lipid that complexes with the mRNA, 2 more lipids, one of them pegylated to structure to the LNP and to give it some stealth abilities, so it can be circulate in the body for a little prolonged time, and usually cholesterol, because this gives us some rigidity into the membrane of the nanoparticle. And frankly, the formation process for LNPs is pretty straightforward. We have lipid components that are usually soluble in ethanol. We have our payload, oligonucleotide, mRNA, whatever, which is soluble in water. If we mix this in the right ratio, and you can even mix it just in an Eppendorf tube by shaking it, you will form some kind of nanoparticles. So for us, as someone who's developing manufacturing equipment, the trick is to do that in a very controlled manner, so we end up with good quality particles. There's a few top-down approaches to do that, where you extrude such a suspension or you sonicate it. These come with quite high load onto the payload and onto the particles. Therefore, we prefer the bottom-up approach here, which was traditionally done as a thin film hydration or even manual mixing, what you would do on a lab scale. But there is a couple of systems based on microfluidics that allow to automate this process to a certain extent. There's differently shaped mixers to do that. And there is, I would call it macrofluidic devices here, the famous T-shaped mixer, the impinging jet mixer. This is what we are using. And there is also fluidic oscillator approaches to manufacture LNPs. While, of course, manual mixing is something for R&D only, the microfluidics, and then even more so, the microfluidics are systems that are also suited for upscale manufacturing. Little look on the market. We are in 2025 now, around 60 billion estimated growth rate until 2034, up to 250 billion. So there's quite activity in this market. And you also see that besides North America, Europe is going to have, or is projected to have, quite a share in this development over the next years. And if we look into the modalities, so the different targets that are currently in clinical studies, or even also preclinical studies are included here, we see oncological disorders, we see infectious disease, of course, we have genetic diseases and so on. So that's quite a broad field of applications. And we also see, and I said that in the beginning, when I talk about our mRNA, that's not entirely correct, because there's different modifications of that. There's small interfering RNA, there's even DNA you could use, and there's other payloads like the CRISPR we also heard before, that can also be delivered via LNPs. Still, mRNA has a major share in this currently. If we look into the different targets, we see something interesting. I mean, infectious disease is a mass phenomenon, of course. Again, the COVID vaccine, this was something for the whole world, basically. But if we talk about cancer, if we talk about genetic disease, this is something that is more patient individual or shared by a relatively small patient group. So next to the conventional therapies, where we have this one size fits all, one vaccine for the world, we are increasingly more looking at applications where we either have small patient groups that, for example, share a common tumor marker, or even individual patients that have very specific problems that we want to solve with our treatment. And this is something we want to reflect in our portfolio. As said, we see huge potential in this for the next years. However, there is a few problems in current manufacturing, in our opinion. One is that scale up in conventional technology can be an issue, and I will come to that in a minute. but also that there is, to our understanding, not much solutions for individualized or small patient group applications in the market currently that are suitable for GMP and that would allow us to quickly access clinical trial material in relatively small batches. So we're trying to solve this in Leon, and basically we based our strategy on 2 pillars. One is the FRJet mixing technology. This is the modular mixing system that is basically our major technology. And the second pillar, of course, is the equipment we have built around this mixer to address different stages of development from R&D to GMP manufacturing. First, I want to give a little insight into the geometry of this modular FRJet mixer. So what you see here, this is the internal geometry of the mixer. You see it vaguely resembles a T-shaped mixer, but there's significant differences to that. So we have 2 starting material streams coming into a dedicated mixing chamber, which is perfectly spherical, which allows us to establish a very homogeneous mixing regime in there. We have the flows entering the chamber via what we call pinholes. So they are pretty small in size, can go down to 100 microns. This allows us to have very high energetic influx into the mixer. And both these pinholes, but also the central chamber, come in different sizes. And this is what makes this a modular mixing system. And this allows us to control the process way better than you could do with a static mixer or with a single geometry. This is how the FRJet usually would come. On the one hand, we have a reusable metal version of the FRJet where you can see this geometry that I explained. It comes basically as a puzzle, different core pieces with the spherical chambers, different inserts for the pinholes. Here we have a single-use version with the very same geometry, but this is something we're using in our small batch GMP device. I will come to that a little bit later. And down here, we see what happens inside the mixer. And of course, it's an impinging type mixer. Our 2 starting materials impinge in the middle of the mixer. And over a very short time, a very stable and highly turbulent mixing regime establishes inside the FRJet. So what's the advantage of that, and what is the advantage of the modularity? If we look at traditional mixers, let's say, I have representations of a Y-shaped microfluidic chip here, or a T mixer. It's known as the impinging jet mixer as well. The intensity of our mixing regime solely depends on the flow rate we put through the system. Yeah, because the geometry is fixed. So the higher we go in flow rate, the more mixing intensity we get. And mixing intensity in many nanoparticle formulations, especially in polymer ones, directly correlates with particle size. The more intense the mixing is, the smaller your particle size gets. So basically, if you use one type of mixer, you have very limited control about particle size when you need to increase the flow rate during scale-up of your process. And this is where the modularity come in. Of course, for one configuration of the FRJet, we have the increase in mixing intensity with flow rates, but we have differently sized cores that allow us to dissipate the energy over different volumes, and we have differently sized pinholes that allow us to bring down the mixing intensity if we increase flow rates. And this allows us to do 2 things. We can control particle properties within the boundaries of the formulation, of course, by using different configurations of the mixer. But it also allows us to replicate the same mixing regime at different throughputs. And this is what makes this system very suited for process scale-up, because we can replicate the same mixing regime at different scales of the process. First point, controlling particle properties. This is data from a polymeric nanoparticle model that we created at a single flow rate. This was 10 ml a minute, so fairly low. And we used more than 60 configurations of the FRJet to produce the same formulation nanoparticles. And we get very different results in terms of size. But this is not chaotic. This is systematic. From left to right, we see increasing mixing intensity that we achieve by using smaller and smaller pinhole inserts in the mixer. And we see color-coded, the larger our core becomes, the more energy dissipation we may have, the larger the particles again become. So instead of one mixing regime with a static mixer at this flow rate that creates one size of nanoparticles, we can create all these by purposefully selecting a specific FRZ configuration. And the second point I mentioned was we can maintain mixing intensity during scale-up. Here we went through the full scale that we can do with our equipment from 10 milliliters a minute up to 1,000 mil per minute, so 100-fold scale-up. And we selected different configurations of the FRZ here, and we were able to maintain particle size very consistently over this whole scale-up. And of course, this is something you need when you want to scale your process from R&D to GMP production. You need to increase the throughput, but you don't want to change the particle properties, of course. A few examples. This is data from a study we did with lipid nanoparticles based on SM102. This is the formulation known as Spikevax from the COVID vaccines. And here we just tested different process parameters, let's say. Here, left to right, we see low total flow rates, medium and high total flow rates. We worked with different concentrations of the starting materials from low to high, and high basically means the upper limit because of the solubility of the lipids in ethanol. We use different buffers after dialysis for downstream, basically to create different conditions for the nanoparticles to mature. Anyway, overall, for all those process parameters that you may want to change in your process development, we obtain very consistent results in the nanoparticle quality in terms of size, in terms of polydispersity, but also very high encapsulation efficiency, so way beyond 95–96% of your mRNA actually ends up in nanoparticles, and also the recovery, so looking at the full process recovery, especially at high lipid concentrations, we are close to 100% of recovery of the payload. We put these into an animal study, of course. We were able to confirm good transfection efficiency from these nanoparticles to the animals, especially for highly concentrated LNP solutions, or starting material solutions, by the way. So overall, again, the takeaway is that we have certain freedom in the flow rates, in our concentrations. The mixer itself is rather agnostic to these changes in process design, and that leaves it up to you to design the best process for your replication. Another, maybe a little bit, how should I say, special example. This was a collaboration with Serum Biotech we had 2 years or 1.5 years ago. They wanted not to encapsulate mRNA, but plasmid DNA. And they wanted to encapsulate pretty large plasmid DNA up to 24 kb, which almost comes into the range of size range of the lipid nanoparticles we wanted to obtain. And this is something we hadn't seen before, that this could be done in lipid nanoparticles. And this has really, this can only work if you have very intense and homogeneous mixing regimes. Long story short, again, with this mixer, we were able to encapsulate even these large pDNA into lipid nanoparticles, and still achieved pretty good results in terms of encapsulation efficiency and polydispersity with these as well. I want to close that first section about the mixing technology as such and talk a little bit about the product portfolio. We have developed, this is 4 devices, although I have to admit that number 1, the nanoscreen, is still in development. We hope we'll have that soon. Basically, we developed these to cover this whole range of total flow rate I talked about, 10 to 1,000 milliliters a minute. We have 2 devices for process, or one for R&D, one for process development, the nanoscreen and the nanolab, and we have 2 devices for the GMP. One is the NanoMe, and one is the NanoUs. The NanoLab is for process development. Say you have defined your formulation, now you want to tweak it, you want to find your process parameters. You want to go through different iterations of the FR-jet reactor, you want to test the influence of that, and you want to scale the process up to 400 mil a minute, for example. This is what you would do in a NanoLab. And for the sake of time, I'll keep that short here. We have the NanoLab in the exhibition. If you're interested, feel free to step by, and we can talk about that in more detail. I would like to come first to the NanoUs. And this is intended for large-scale production of, for example, prophylactic vaccines. Here we're talking about hundreds, thousands of liters of product in a continuous manner. This is clearly the point where we were very happy about the collaboration with Harro Höfliger, because building such machines is clearly something Harro can help us a lot with. And also because we choose the approach to build this machine on a build-to-order basis, because we want this to be customized. Basically, in this machine, this can work aseptically, this can have automated cleaning and sterilization routines, this can integrate process analytical technology. There's many different options that can be established for more or less a specific process or a process-specific customer and his respective needs. So here, this is clearly for large scale. And finally, I want to spend 2 more slides on the NanoMe device, because this is something completely different. What we have done here is we targeted, or we designed it for smaller batches. This could be clinical trial material, this could be stratified patient groups, this could be even individual patients. We took the whole fluid path into a disposable system. This is, to our knowledge, the only system that can actually do this. And we did that by coming up with a 2-fold system. One part is the benchtop device. This is what does the control, the actual pumping and so on. And we have a disposable cassette, how we call it. So basically we have bags for our starting materials. We have our disposable polymeric FR-jet mixer in here, and we have bags for the product and a little bit startup waste. This is on the back side of the cassette. Also the cassette is at our exhibition booth here. And what the benchtop device basically does is it closes around the lower part of the cassette and applies controlled air pressure onto the starting material bags. And this is what does the actual pumping. And the benefit of this is we have no contact between the machine and our starting materials or product, which means that once a process is finished, we can take out this disposable system here, detach the product bag, give it to downstream, discard the rest, and the machine is ready to go for the next batch. And this allows us to produce, we calculated, up to 140 batches a day, theoretically, with such a machine. I mean, this could be 140 patients. This could be 140 different materials. Yeah? So a batch a changeover between different batches basically can happen in no time. And since the whole system is closed and even enables aseptic processing here, the bags can be filled through filters and so on. We have much less requirements regarding clean room environment with this machine. And overall, this of course allows us to bring down process time and therefore also manufacturing cost a lot for such a system, if we are talking about not mass market supply, but smaller batches actually. And with this, I just wanted to put on the product portfolio once more. Nanoscreen, as I said, hopefully coming soon. NanoLab for process development, and then the NanoMe covering clinical trials and small to mid-sized batches. And finally, the large-scale commercial over the NanoUs. And with this, I want to say thank you. I'm open to questions, of course, and wish you a nice lunch break then. Thank you.